כן, אני מכיר אותה. ארן שם.
מה להמשיך? כדאי להם לשמוע.
סתם. אני לא יודע אם הם יבואו או לא, בדרך. את האמת, בדרך לפעמים, לא... בחלק מהמקרים אנשים שותים. או בנוסף, אז זה יתפרסם שלוש פעמים. יש אנשים לפעמים, הם אומרים, אם רוצים לדבר עם ה... לדבר איתך שתקרוא משהו אחר. אני היססתי לשלוח את זה לפעולה, אתה יודע, אמרתי לו, בגלל המצב במיוחד בשבוע האחרון,
אף אחד לא...
לא, יבואו, להבין שלי בטוח יבוא. הוא בטוח יהיה.
נותן לך,
כן.
באנגלית.
לא, הפיצ'י שלי אמרתי לו לבוא, כן. אבל את האמת לאנשים אחרים, הרגשתי, אם הם לא רוצים לבוא, אמרתי, שלא יבוא.
לא צריך לדחוף.
לא רוצה לדחוף. שלא ירגישו שאני דוחף.
תראה, לא ציפיתי שיהיה פעולה.
את האמת, אם זה היה ננו גון, יכול להיות שהיה הרבה יותר בגלל שהקבוצות, שם נותנים את זה פעם בשבוע, והקבוצות די, אתה יודע, מחייבות לבוא להרצאה.
יכול להיות שאז היו כועסים.
יכול להיות, כן. יכול להיות. קשה לדעת בדיוק ככה. תגובות.
לפחות לא ביקשו לפתר.
כן. לפחות על זה.
אני גם שני אנשים.
לא, בטוח. לא, אנשים בדרך כלל באים בדקות האחרונות.
כן. נותן לך את זה פרטי.
כן, אני לא שמעתי, אף פעם לא שמעתי. דרך אגב, בסנטה ברברה עבדת על מייקרוטובולר.
אני עדיין עובד על זה. אבל זה לא במקום, זה עבודה אחרת. זה סגורה.
How are you? How was your trip to?
It was nice.
Nice. Uri, Hussein. We were just. I was describing a little bit the desalination work that we are doing. So he's doing the simulation for that.
Nice to meet you.
So the to Germany was
It was
okay.
nice.
It was nice. And the Germans.
Yeah.
Also nice.
Different. Yeah, it's also nice.
Yeah.
They have nice things.
Okay, so it was
Some bad things also, like delay of the trains.
yeah, that's it was not like this 20 years ago, but now it is.
It's nice.
It was good.
It was about HPC.
Yeah, so HPC, it's what it stands for?
No, HPC like the cluster.
High performance computing.
High performance.
Okay.
Nice.
You got you presented a poster?
Yeah, I did.
People were asking questions?
Yes, they asked me questions. Yeah, and then I found some of them are also working on ionic liquid long time ago, and they asked. But what they did is quite different than what I'm doing. And also, I got like someone collaborator having a name Ronen, like your name.
Okay,
And, yeah.
that I guess from Israel.
Yeah, he's in the Shiba or
Oh, Ronen Gonen, maybe. Gonen, maybe this last name? No.
Ronen Bergovich.
Oh, Berkovich. Oh, I know him. Yeah. He was here a few years ago. Yeah.
He's doing AFM.
IFM, yeah, exactly.
He's a good guy.
Yeah.
But Ronen with A is a typical Irish name.
Also Irish, yeah, it's exactly, yeah. We write it with E, so that's normally Israelis, but with A, it's Irish, yeah.
Also found Ronen like but he's Irish.
Yeah, Ronan maybe.
What did you study? Physics or?
Yeah, I studied physics. And now chemistry. More or less like, what do you say it? It's physical chemistry.
chemistry.
Yeah.
Oh, yeah.
Yeah, it's
Maybe
few minutes.
there is also some
Defense, yeah, I know. Yeah, of ball.
Maybe they did not complete.
Yeah, well, PhD defense is very long here because there is long question sessions that can take hours.
Obviously, that they.
Yeah, no, I don't know if everybody.
Yeah. Even for you to implement something. It's better. Where did you study your undergrad?
on both the undergrad and the master's in the European science.
I was in Kenya in Uganda.
A long time ago.
I don't know, we normally wait a few minutes after the 12 o'clock, no? It's that's sort of the custom.
Yeah.
Okay, you know, I think you were talking by email, you were Uri and David the Sancho.
Thank you.
Hola.
When did you move to Europe? When did you move to
I mean, I
graduate?
moved. Okay, I moved here like 2 years
2 years
ago.
ago. 2 years ago?
Yeah. Yeah.
I guess we should start. They say it's always a pleasure to introduce somebody that you don't even need to look at his CV in order to introduce him. This is because we both started the academic career together at the Hebrew University. We studied 3 years chemistry. Yeah, if nobody's coming, please. As a student, the only thing that is on your mind is what are your grades? And for the 3 years that I've been of that program, we had the highest GPA, grade point average in chemistry, and I think one of the highest at the university. So that was quite, I don't know how he did it, but he managed to do it. So, and after that, he did a PhD at the Weizmann Institute. He was using atomic force microscopy to look at properties of material that have only thickness of only few layers. I think by any standards, one can consider his PhD as a success because in his list of publication, he had 2 Nature papers and 1 Science, all as first author. That's also, I was startled then how he managed to do it. After that, he got a Marie Curie fellowship, went to UC Santa Barbara, and then he worked on supermolecular assembly, especially microtubulus. And after which he got a faculty position at the Hebrew University, and which is right now a full professor. A few days ago we were talking, and you mentioned that you hate when you listen to talks that you need to wait with your question until the end of the talk. So we will practice it. So whenever you have a question, just fire us in.
Exactly.
So
Thank you very much, Ronen. Great pleasure to be here. And indeed, please ask anything, and I will not go fast. I promise. I like actually questions during the talk. If you feel that any term is unclear, I'll define it and explain everything. Okay, so the challenges, there are 3 challenges in our lab that we try to address. And one of them is to resolve the structures of large macromolecular assemblies that form in solution. The second challenge is to determine what are the intermolecular forces that lead to these structures. And also to understand, the third one is to understand the dynamics of how the structures are forming when we take the subunits and allow them to assemble to form these large structures. And so we study the kinetics and dynamics of self-assembly. And there are 2 kinds of assemblies that we study, one that can attain equilibrium, and the other one that I will not discuss today is related to the tubulin and microtubule, is the case where the assembly does always operate out of equilibrium. This is more complicated, but I will not address it today. So I'll discuss today the case of virus capsid assembly. In the life cycle of a virus, this is the laser. Yeah, can you see it? First of all, half of the known viruses are icosahedral viruses, icosahedral. And during the life cycle of a virus, the virus has to enter the cell, then disassemble into its component, and then use the machinery of the cell in order to multiply its subunits, element that creates it, and then eventually select the correct subunits out of the entire many molecules that exist around it, and assemble the capsid in the virus itself. So usually the capsid of a virus contains many copies of one or two proteins, and it encapsulates the genetic materials that forms the virus, that contains all the genetic information that is required to create the virus. And icosahedral viruses has a triangulation number that correspond basically to the size of the virus and to the symmetry that it creates. And the larger the T number, the larger the capsid is. So 3 or 4 correspond to the number of 60 times the T number is the number of subunits that creates the capsid. Okay, so this is a brief introduction to general icosahedral viruses. And the one I'll talk about today is the hepatitis B virus, or HBV. And this virus, hepatitis B there is a vaccine, but the chronic infection leads to the death of about 1 million people a year. So it's still something that requires treatment and the development of antiviral drugs. The capsid can assemble around the RNA and eventually become later a DNA. But it turns that 90% of the capsids in the cell, 90%, are actually empty capsids. So the assembly, and they are used as a storage for later maturation of the capsid. So the capsid, virus capsid, is a promising target for antiviral agents that eventually people are still investigating and want to, and addressing the mechanism of self-assembly can be useful for this kind of goal. So, and the question we are, so in general, also the assembly of virus-like particles is used for other purposes, like protein nanocapsules. People usually use capsids to encapsulate all kinds of materials or genes, sometimes for gene delivery applications, or usually also they can be used as subunits to create three-dimensional materials, more complex materials, or as a scaffold for displaying antigens for vaccine applications. So in all of these cases, the structure, the dynamics, and stability are relevant questions. And so the case of hepatitis B can be, yes.
You said you wanted a question.
Yeah.
So I would like to challenge this idea of like the capsid being a good target. Does the sheer amount of protein that has to be generated not involve that you would also have to provide a crazy amount of the drug that you're providing to assemble? Because you have so much of the capsid protein, then you would need too much of the drug, and then that would make targeting that specific bit of the virus difficult.
Well, that's true, that's correct. However, maybe I'll show you today the approach is not, the best approach is perhaps not to prevent the assembly of the virus, but perhaps to lead to kinetic traps that will disturb its functionality. And that can require much less of the drugs. So this is one of the conclusions that may come out of this research, actually. Okay. So the assembly of hepatitis B virus can be recapitulated with purified proteins, of the capsid proteins. And it turns out that if we take the capsid proteins and increase the temperature or the ionic strength, both of them can lead to either screen the repulsive interactions between the negatively charged dimers. Usually the subunit is a dimer, not a monomer. So this is the basic subunit, dimers. And 120 dimers forms the T equal 4, which is 240 monomers, and the size is 33 nanometers in diameter, or in vitro. We also see and in vivo that they coexist with T equal 3 capsids that has only 90 dimers or 180 monomers, and the diameter is 28 nanometers. And so increasing the ionic strength or screening the electrostatic interaction can help the assembly. Also, the temperature may expose hydrophobic parts of the protein and also lead to enhance the assembly. So this kind of system can be recapitulated in vitro. Okay. And then the question is really what happens when we start with the monomers? What happens in the middle on the path for the assembly of the full capsid? What are the rates? What are the intermediate structures that form under what conditions? And this problem is very challenging because there are 10 to the 30 possible intermediates on the path from the dimer all the way to the full capsid.
How did you get to that number, by the way?
Oh, so we did Monte Carlo simulations. I'll explain soon. And we eventually evaluated this number based on these simulations. I'll show you soon how we did it. And one of the conclusions that I'll show you is that to properly assemble the capsid, we need actually to maintain weak protein-protein interactions. Because otherwise, if we increase the strength of the interaction, and not by a lot, we can lead to kinetic traps, actually, very easily. So the way we actually investigated the assembly process, the final product is using solution X-ray scattering. In this technique, what we do, we focus an X-ray beam onto a sample. This is the beam. We focus it here onto a sample, and then the sample is maintained in solution. And the scattered photons are then collected onto an area detector, two-dimensional detector. And the typical pattern that you see is that of rings, because we have all the orientations in the solution. And unlike in crystallography that you see spots of diffraction peaks, here you see rings that we then integrate azimuthally each ring or the intensity. And then we get the one-dimensional curve of an intensity as a function of the scattering vector or the scattering angle, if you want. And then we need to do data analysis. And the advantage of this method is that it's simultaneously done in solution, it's label-free. There is high spatial resolution. You can get the wavelength of the X-ray is basically 1 angstrom, and you can go up to very large dimensions of about 100 nanometer. The temporal resolution can be 20 millisecond and up if you go to synchrotrons, and it's a highly sensitive bulk method. However, there are challenging. The data analysis is challenging because we measured the scattering intensity rather than the scattering amplitude, which correspond to the famous phase problem in crystallography. It's the same problem in solution. But in addition to this, we also average. We have all the orientations, and we get one-dimensional curves. And we're trying to infer about three-dimensional structures, which means that we lose a lot of information. So the way to address it is to compute what we think, a model that we think is the structure, and then compare the scattering and compute forward based on some model, a geometric model or some atomic model or whatever. We compute the scattering curve that we expect, and then we can compare to the data. So we developed software to do it. The first software we did was developed about 10 years ago, 15 years ago, actually, where we can model geometric structures like spherical or cylindrical objects with multi-layers and different density, radial densities, or layered structures. And then we could also place them in lattices and compute scattering curves. And this was insufficient, so more recently we developed a more advanced software. It's called D+. This is a large project, much more advanced software where in this case we can take a subunit. It can be either a geometric model or an atomic model, and we can place it in an assembly symmetry which has the information of how many copies of the same subunit are shifted and rotated in space. And that can be done in a hierarchical manner for different subunits and different symmetries. And eventually we can compute, and like Lego, we can construct structures at different resolution and compute the scattering curves and compare with our data. Okay, so this is the technology that we use to to analyze our data. And here is an example. The two extreme cases related to our case is the dimer. This is the dimer, is the dimensions of 2.5 nanometers. The blue symbols are the scattering data. And the red curve is the atomic model that was available in the literature for the dimer. And you can see how that can fit the scattering curve, the measurement. On the right hand, yes,
sorry,
yes.
I have been trying to use this program a lot.
D+?
D+, yes.
Oh, yeah.
But
Okay.
I don't have atomic models for my structures, so I don't know how. So how
You can use geometric models as well.
So can you not use like an ensemble of spheres?
You can. The way to do it is to add the size of the sphere, or you can use multiple sizes of different spheres, and then you can add a list of positions and orientations of the geometry. If it's not a sphere, it matters.
Yeah.
If it's let's say a slab, then the orientation matters. If it's a sphere, it doesn't matter. So just the positions, and you can add it. It's called you can add. I can show you how to do it.
And then I have a philosophical question about this, in particular for this kind of forms that are more like elliptical.
Mm-hmm.
If you want to compare, so then when you have this real space model for your Fourier transform, if you want to compare several samples that they change in size, I don't know, or in thickness, or then you come across the problem that you have to then do analysis of the real space model, right? You
create a model, a model that you think what it is, okay, and then you compare with the data, and you try to fit maybe the dimensions of the radius or the thickness of the.
But then you have to quantify in the real space structure.
Yeah, you need to define what are the dimensions of the structure.
So yeah.
So you need to know something about it, or to guess something and then fit it. But not too many parameters. If you have too many unknowns, then you'll definitely fit everything.
Because I see a clear advantage in the right-hand side one, that you have a sphere that has a motif in the surface.
Mm-hmm.
I can see a real advantage in resolving the motif outside, because I think maybe those peaks at high Q are related to that.
Yeah. It is.
But in the left one, which
Mhm.
is an ellipsoid with I don't know, like it's more a question whether it's worth to do the
If you have the wrong
modeling or just
you have an incorrect, so you ask what is the resolution basically, what the sensitivity, right? How sensitive is it to the structure itself? If I had a slightly different structure, whether I could still fit the data.
Yes.
So it turns out that even, I mean, if you have a slightly different structure, you may see a slightly less good of a fit. So it depends. I mean, for example, in this case, we also took the hydration shell into account, which has a slightly higher density than and it matters. But it is true that when you talk about small features, then it depends on the quality of your data. In this case, for example, it is indeed a monomer, a dimer, just a dimer. Sometimes you have, in addition to your dimer, you have some other things, and they may also be there, and it depends really on the case. For example, one thing I can tell you is that if you have the incorrect structure from AlphaFold, sometimes you will see that it doesn't fit your data.
Exactly.
Yeah. If the structure is incorrect, you can definitely, what you can do for sure is to exclude the models, okay, that are incorrect. If you have two models that fit the data equally well, then it means that you cannot resolve between them.
Yeah.
That's something that is an issue. You need to be careful there.
You need to know
You need to know more than, and we do a lot of cryo-EM to accompany our studies. It's not the only method we are using.
Okay.
But here on the right side, what we did is we took the same dimer and put it in the icosahedral symmetry of T = 4, and then we computed this model.
No, that's great, because the one in the right, there is no other way to do it.
I'll show you later
Okay.
what can be. There are actually other ways to do it. Okay, I'll show you. So, and here is, so this was the two extreme cases, okay? Then the other thing is that we did a whole phase diagram where we started at zero, we did it in 3 temperatures and at different salt concentrations. If we take the same capsid protein and now increase the salt concentration, you see that you go from something that looks like a dimer to something that has the features of the entire capsid, okay? And you do it at different temperatures, you see, as you increase the salt concentration, indeed you have more of the capsid, but what exactly is going on here, this is more challenging, okay? What are exactly the possibilities to fit such data is more complicated, okay? And how did we address it? So the way to address it was to map the phase of the possible intermediates that can be created on the path from the dimer all the way to the capsid. So what we did Monte Carlo simulation. In fact, we did umbrella sampling of Monte Carlo simulations. And we represented the structure of the entire capsid using graph models, if you know, graph representations. And then what we did in the simulation, we took different windows. Let's say we started from an intermediate size. So this is, eventually we can characterize the intermediate on the way from capsid to from the dimer to the full capsid based on the degree of connectivity, which means the more compact structure, the higher the degree of connectivity. This is this axis. And here is this axis is the number of dimers. So here we start at one, and the last one is 120. And the map represents the number of possible intermediates at each point of this representation, which means that if you have 80 dimers, for example, this is highly populated states, because and the degree of connectivity is, let's say, 0.3, more or less. This is the highest, this is the most populated state, which contains something like about 10 to the 28 possible states in this kind of structure. Okay, so there are many ways to create this, and this was done by estimating these numbers, was done based on adding or removing a subunit with no association energy between them. It's zero energy. We just did simulations where we added the dimer or removed the dimer, make sure that we count only the unique intermediates, and we did it until we converge, more or less the simulation converged to get the density of states, more or less, okay? So it's of course, it's impossible to cover everything, but at some point the simulations converge and the distribution converge. And you can normalize it based on the known solutions at the edges of this distribution, because you know that for 1 or 2 dimers, you can calculate what the numbers should be. So you normalize the distribution and everything can work like this. And we compared it with the distribution of t equal 1, which can be solved analytically. We did the same kind of density of states. And then we created a representative library of these states, maybe a few thousand. And we computed the scattering curve of each one of those representatives in the library. However, still to fit such data, the one I showed you, there are many ways to do it. So it's an ill-defined problem. And the way to do it is actually to count the stability of those states. Because the holy states, the one with many holes, will be less stable than the one with more compact. That are at the same number of subunits, if they have more contacts between them, then they are more stable, and they will basically dominate the distribution of states that are likely. Okay.
How can you count? How can you account for stability if you don't have association energy?
That's the parameter you fit to the data in order to recapitulate the scattering rate. So this is
okay. So you are using the
experiment.
Okay.
Yeah, you're using now the experiments. Now you see all these curves were computed based on the library of states, and then we took the Boltzmann weighting of those states according to the association free energy that was fitted to the data. This term here is the association energy, standard association free energy per contact, okay? C is the number of contacts in a state, and then the omega is the number of states that represent the same kind of size of an intermediate and the same degree of connectivity of an intermediate. So this is the degeneracy, if you want, that comes from the simulation that I told you about. And then this is the free energy of this assembled state. And you need to counterbalance it with the free energy of the dissociated state, which is the last term, okay? The last term is the number of subunits when the entropy of this is the entropy of the same number of subunits if they are totally separated, okay? Because this is the balance, the assembled versus the disassembled state. So this is basically the free energy difference. And then this determines basically the mass fraction of a state with S subunit and C number of contacts, okay? Yeah.
One last question.
Yeah. No,
In
not
the-
last. Why not?
In the intermediate states, how do you account for polydispersity?
Okay, so you basically, okay. So what you do in intermediate, there is, so you have representative, it's actually, it's in the question phase. What we did clustering algorithms. We applied clustering. We computed all the states. And then, in fact, we took the centroids of this cluster of states for the same size and connectivity that we took it, basically. Okay, so we had a representative scattering curve that based on the noise in the data, we accounted for a whole domain, actually, that correspond to the same. It was a representative all this, not even one size and one degree of connectivity. When you go to the highest number of, when you have more than, below 60 dimers, each, you have, they are more separated, but once you go to the highest numbers, I'll show you in the end because it's the end of the presentation. I'll show you the clustering map that we did, how we actually picked the centroids of these maps. So we have a good representation of this, okay? But then the parameter you fit in order to fit all this data is the free energy of association between subunits. And this determines the distribution of those representative states. And that's the only thing you fit to the data. And that's how you actually get all these curves to fit at each temperature. And so the free energy changes as a function of salt, okay? So what are the results? The results actually are very intuitive in the end, as you'll see. It turns out that 99% of the mass is found in either the free dimers or the full capsids. There is a little bit of 2 and 3 dimers as intermediates. It turns out that the less stable complex intermediates actually dissociate in favor of the most stable structure, which eventually, if you think about it, the final product must be the most stable one. And it's in the exponent. The number, as you increase the number of contact, it's in the exponent. So this is why they so dominate. These states are so dominant. So if you now change at each of one of the temperatures, you can see now the distribution of intermediates that form the capsid as a function of the salt. You see that as you increase the salt, you have more of the T equal 4, you have a little bit of T equal 3, and you see the decrease in the mass fraction of the free dimers. So in the end, it's not so many intermediates that actually form the final product. That's what happens at higher temperature. So this was one of the conclusions. And then what you can do, you can actually plot how the free energy between subunits is changing as a function of the screening length, which is the inverse 1 over the square root of the salt concentration, which means that at long screening length here on the right, you have the low salt concentration, where you see no assembly. And as you increase the salt concentration, you start to see assembly when you cross the critical concentration. And then if you have too high salt, you start to have malformed particles, which you can see in the microscope. Here at the bottom, when you have higher salt concentration, like half a molar or so, then you start to see these are intact at the top here. These are better, nicely formed capsid. Here, you can see that they are malformed already. So by increasing the salt concentration, you change the standard association free energy between subunits by about 2 kilocalories per mole, or 1 kT, more or less. And then that's enough to go from no assembly all the way to malformed particles.
You change it by increase it. You increase it.
Yeah, you increase
it, and that's why you have
malformed.
Okay.
Yeah,
Or
you have kinetically trapped states that are not forming well. Okay. So this is the case for the steady state. And now the question is what happens on the path, on the assembly. Okay. So here, what we did, we used time-resolved X-ray scattering. Okay. Using a stop flow apparatus where you mix the capsid protein with salt solution. Because salt, you start at low, here you don't have salt, and here you add salt as much as you want to initiate the assembly. So at the mixing point, there is a mixing point here, and then the solution goes to the measurement cell, and then you follow it as a function of time. You can then start to have scattering curves as a function of time after the mixing. So the 0 time is your mixing point. Then what you get, sorry. Okay, so now, let's see if, to analyze, now we have more data. Sorry. Yeah, it's okay. Now to analyze this kind of data, now we have multiple curves. And you have, actually, it turns out that you have to use another technique in order to reduce the bias of your fit, okay? So this is called maximum information entropy optimization. It's a method that if there isn't now a distribution of states that you want to fit to your data, to minimize the bias, you maximize the information entropy, which is given by this term. You maximize it subject to the constraints that all the sum of the distributions should be 1. All of them should be positive. And eventually, the signals that you see, the distribution of that you have, should fit the data. Okay, so these are the constraints. And this leads to the broadest distribution that the data do not exclude. Okay, this is a methodology that we apply. But this was insufficient because there is one thing that you need to take also into account, which is the prior distribution. And it turns out that this is another way of biasing your fit. If you don't take the correct, you can get more than one way, you can fit your data in more than one way. So the correct way to do it, we think, is to start from a distribution that is weighted, that has some energy, association-free energy, to start with a distribution that is slightly below the critical energy that is required for assembly, which will give you the reactant-like state, okay? Or the product-like state, an energy that is sufficient to create the product. So you go to the last equilibrium state, and then you can go backward in time and start using the distribution of the earlier data point to go forward in time, or to go backward in time. You take the distribution of the latest data point and go backward in time. Did I explain myself? It's either you always take the result of a fit at some time point and use it either backward or forward, if you go backward or forward in time. So you start from the last data point and go forward in time. You start from the first data point and go backward, forward in time. And you always take the prior as the result of the fit of your earlier data point. Okay, so these are the two cases where you have, this was critical actually to get physically relevant results in this case, okay? And the difference between those two pathways gives you the error of the distributions. So that's what we did. Here I am showing two conditions, mild conditions that correspond to a salt concentration that is more or less physiological. And this is the time result data in the case of where you have higher salt concentration where the assembly is aggressive, more aggressive. When you look at this data, you see that the time, so this is the first data point is after 20 milliseconds or so. And then you see in the case of the mild condition, you see maybe the first 10 seconds, not much is happening. And as you start going in time, up in time, you see more and more assembly. Here you see actually much faster assembly. After a quarter of a second, you already see a lot of assembly. Okay. And here is another representation of the same data. You see here in this data, at the mild conditions, when you plot the scattering curves on top of each other and the black curves at the feet, you see that actually you see isosbestic points here where the curves cross each other. And that's an indication of a two-state system, okay? So what it means, what it suggests is that the intermediates actually do not accumulate to a significant amount. Okay, so when you now do the analysis and the full analysis, you see you can plot the mass fraction or as a function of the number of dimers and at different time points. This is the axis here. And you see the lag phase, and you see the depletion of dimers and the formation of, you start to see 2 and 3 dimers, and then the formation of the final product. That's the main thing you see in this data. That's another representation of the same thing, of the same data. So within our resolution, which is about plus minus 2%, intermediates did not accumulate under mild conditions. Okay. Now, when you go to aggressive condition, you plot the same data at this representation, you see no isosbestic point here, you know that you have a different mechanism. When you do the analysis, you see fast depletion of the monomers, you see the formation of intermediates, all kinds of intermediate between 7 and 35 dimers. You see the formation of T equal 3 and T equal 4 very quickly. Okay. And here you see the formation of intermediates and their disappearance. Okay. Now what you can do to rationalize this, that this is an intermediate conditions where you see decamers actually that happen to be a free energy minimum as intermediates. Now to rationalize all of this, what you can do is, because we have all of these states, we can actually, and we know what is the standard association free energy for each of the conditions, we can plot the free energy landscape of a particular state or assembly conditions. So if the interaction is too weak, when we plot it below the critical standard association free energy between subunits, what you can see in this energy landscape, this is again the degree of connectivity, and here it's the number of dimers, and here now it's the free energy of the system, the grand canonical free energy. Here what you see is that the high energy is here, this is the higher energy, but you can see that there is no way to cross, the lowest energy is really at the monomeric state or the dimeric state. Okay, there's no way to cross the barriers to create the assembled bars, capsid. When you have mild conditions, this is the energy landscape. What you see here is now the minimum. You do have you can form capsids, but also you can see that the minimum free energy path is going through the highest connectivity states, okay? Which means that there is an energy barrier to cross to the structures that have holes in the structure, okay? That are less connected. So there is a very narrow path on the assembly from the dimer to the full capsid, which the system must follow. So it's the broken arrow here, broken red arrow that can only be through this assembly, can only go through this. So the interaction in this case is just enough to form the capsid. And it turns out that if you plot now the free energy, a cut of the free energy through here, you see that there is also a barrier to cross. So if you, this is the sort of a barrier. This number is incorrect. It should be less. It actually should be 17. That's incorrect here. 17 kV. So the barrier is such that you do not accumulate intermediates. So some states either, you see the barrier is actually the reason for the lag phase that we were detecting in this assembly condition. And also the fact that assembly either cross the barrier and then go down to the downhill in the free energy landscape to the full capsid, or completely dissociate and go back to the dimeric state. Okay. So the assembly mechanism is really the nucleation and growth mechanism. Now, if you go to the aggressive conditions, what you see in the energy landscape is that although the minimum free energy landscape is still the most compacted states, there is almost no barrier to go to other assembly states that are more open with less lower degree of connectivity, which means now that you can actually go to all kinds of kinetic traps. The energy barrier also here to cross is much lower. And once you cross the barrier, the energy landscape is going downhill very quickly. Okay. But here, if you go to these open states, because the association is so strong, then the dissociation of these states is more as there is a high energy barrier to dissociate from these states and correct and go back to the correct assembly state. So these kinetic traps lead to the malformed particles that I showed you in the electron microscopes.
So on the two-dimensional profile, where are the traps? Are these the sharp points?
Here.
No, but on the right, you don't have a protection.
You see, this is the cut through here.
Here, you don't have a cut through the.
Yeah, we could plot it.
Okay, and then you see a minimum in the middle. Okay.
There are many kinds. Yeah. And that's a comparison just of the initial state of the free energy between the different salt concentrations. The three assembly conditions where you see a higher barrier and then a lower and a much lower barrier. And once you pass the barrier, it's all downhill. And then if you start in the aggressive condition and you plot the same cut in the free energy landscape as a function of time, what you find is that as you go forward in time, you actually you depleted the monomers or the dimers in our case, and then basically you go back. The energy barrier start to increase. You can see it in the inset here. As a function of time, because you have depletion, now you have the concentration, the effective conditions become similar to the mild assembly conditions because you start to have a higher energy barrier as a function of time, because there are fewer subunits. So it means that the assembly mechanism should follow the nucleation and growth mechanism that we had in the low salt concentration. So concentration or salt can push the system for assembly. And if you reduce the concentration of free dimers, you actually go back to the mild conditions. Yeah. Okay, so the reversibility in the case of the mild condition helps to avoid kinetic traps and to correct for the errors. And in the aggressive condition, the assembly is so rapid, so malformed particles and intermediates can be detected. Okay. I don't know how much time we have.
Oh, well, we are a little bit, that's about the time.
Okay, so I think it's a good point to end. This is, I'll go to the summary. The overall summary is that you have the geometry that determines the number of possible intermediates, which is very, very large, but eventually the standard association free energy between the subunit, making the actual selection of which intermediates will be dominating at each time point along the assembly and in the final product. And the small difference in the free energy landscape of 1 kT in the association free energy can take you from successful assembly to off-path assemblies when the association free energy is too strong. And the students who did this work is Roy Sor, and of course we did it in collaboration with the group of Adam Zlotnick, who provided all the capsid proteins, and we did it at the synchrotrons here in Europe. And these are the funding. This is the current group, and thank you for your attention.
Okay, so there's some
So I owe you
questions.
the clustering. I owe you the slide. I'll show you the no, it's not here. It was supposed to be here. I don't know. I missed it in this presentation. So I can plot it on the board actually for you. Maybe. So if there is a pen.
Maybe there. If not, I would not know. Probably here. Yeah, should be. And this is
Yeah. Yeah. So I don't know if you can see it, but
we can see it.
okay. So if this is the size and maybe degree of connectivity, okay. This would be 0 and 1. So you can now make, we divide it to all these domains, okay? And there are multiple possible subunits in each one of them. But when you then create, what happens is when you compute, for example, the scattering curves, and normalize to the data, it turns out that if we go to the 120, then if at low degree of connectivity, you cannot differentiate between 110, for example, and 120. And as you go to higher degree of connectivity, maybe this would be like 115, okay? Then, so you take only the centroids of this whole domain, which its scattering curves are really similar compared to the noise level of your data. And you take only one that represents this domain. As you go to lower numbers, then the domains become smaller, okay? Then you have better resolution, actually. And below 60, it starts to be that each one can be separated. But still you have a centroid about, because in different degrees of connectivity.
So
okay.
Then so I have then two questions related to that. So one is, if you have a multimodal population, or you are assuming that you have an average structure. So maybe you have a mixture in the intermediate states. I'm just talking about the intermediate states. Maybe you have dimers, trimers, half capsids, and full capsids all together, and then you have a scattering curve that has a little bit of each, or you are assuming one average shape.
So within a domain, it's the average. Okay. Here, let's say this is a domain. You take the centroid. It represents all of them.
Yeah. Okay.
Okay, you don't know if it's really this one or this one or this
Yes.
one.
Okay. And then the other, so then, and this you may, for sure you have said it, but maybe I take longer to understand. So what you're doing is a Monte Carlo simulation to compute a structure that then you feed to your D plus to fit.
So the Monte Carlo simulation, what it does is enumerate we take the full structure, okay? It's 120 subunits. Now in the Monte Carlo simulation, you start from, let's say, an intermediate with, let's say, this part, okay? And then you add a subunit or remove a subunit. You create different structures in the simulation.
And then the one that is minimized to the scattering?
No, just in the beginning, you don't have any energy.
Okay.
You just create the density of states, possibilities of many states, let's say 1 million states.
Okay.
Okay. And then you compute the scattering curve of each one of them, because each one is basically a list of positions and orientations of the subunit, and you can fit it to D plus, and you can compute the scattering curve. But in the end, it's only a few thousands that you really use. Okay, it's a few. I think it's a few or a few hundreds eventually that you really use in the end, because you compute a lot, but a lot of them are degenerative. So by the clustering algorithm, it kills a lot of them. And then maybe a few hundreds. That's all what you're using. No more than that. And they represent the domains that are in all of these phase space, basically. Because the resolution of the data is insufficient to be free.
I understand that.
Yeah.
But still, no, I'm just wondering, it doesn't matter. I'm just wondering about the use for other out of equilibrium systems, for example, because here you just have the one final structure, but if you
Right. Out of equilibrium, we didn't solve it yet.
Yeah.
It's still challenging. It's still a very challenging. We try to use the same methodology, but it's still it's more challenging, actually. Also experimentally to isolate. This thing was possible experimentally, and there it's even more challenging, because even your starting point is already a distribution of states, even at time zero. We characterized it. I mean, a whole PhD was devoted to characterize the initial state. And this was just, here it was simply the dimer. So we apply all kinds of biochemistry to isolate the states and things like this. With size exclusion, chromatography, and things like this. Yeah.
I had a couple of questions in terms of the driving forces for these processes that go into this equation of yours. So at the beginning, you have introduced the fact that by heating the system, you get to induce the formation, and then you also have the salt effects, both of which you explore, and that sort of reminded me to like polymers, like hydrophobic polymers, because I guess that by adding salt, you deplete the electrostatic interaction, so it's hydrophobic driven, I guess. And then the temperature suggests, the temperature effect suggests it's entropy driven, phase transition. So I was wondering about, so where does the entropy of water go into this expression? Is it in the free energy that you're feeding, or is it in the mixing term, probably?
No, it's in the standard free energy is actually including all of this already.
So it's a temperature dependent.
It's a temperature
parameter.
No, it's a salt dependent. I mean, it slightly depends on the temperature, but yes, it slightly depends on the temperature. Yeah, there is. If you see there is some difference in the temperature between the temperatures, but mainly salt.
And the other thing is,
but the standard free energy per contact includes all the effects, including the water release.
And the other thing is, so have you thought about working with mutants so that because you have these free energies that have to be, I mean, in part capturing the contact energy between the monomers?
It's actually
working with mutants to just fiddle around with how much the interactions matter would be very interesting to see whether you can play around a bit. No,
that's a good idea. That's a good idea. Actually, we should.
Maybe nature doesn't do it, and then it's not interesting.
No. It's a good idea. It's definitely a good idea to do. Mutants. I agree. We didn't do it, but we should do it.
About talking about the methodology details, I didn't get how you did the umbrella sampling.
So we divided the space here to different degrees of connectivity, and to different sizes. So you start at a certain window. So the umbrella sampling prevents you from crossing the barriers of it's actually applying a potential that is very, very high if you cross the window. So if the window can be between, let's say, 1 dimer and 10 dimers, okay? And then the degree of connectivity can be between 0 and 0.2. Okay, so anything that crosses these barriers will be prevented. It will not. And the reason to do it is because the phase space is so huge that you have to sample each. Otherwise, you start here, you'll never get here. Okay, so in order to do it, you have to force the initial state to be in one of the domains, and that's how you sample everything in many simulations. It's we did not invent the umbrella sampling, of course.
Yes.
It's very common.
Yes. Another maybe a quick one. So I guess that, well, obviously viruses work by putting DNA or RNA, inside the capsid. So how does that perturb your model of how things work? Is it they're just encapsulating whatever's in there in the cell, or is there like a different? It's a different thing altogether when they're functioning.
Very different. It's going to be very different. But I think some of the principle may still, because once you have RNA, then it can be that RNA will simply attract more subunits. So it could be more, it could be a different mechanism of recruiting much faster all the elements attract. It's like it will require another approach to address it. But also, I mean, experimentally, you'll need to control the system. This system was well behaving. The RNA system, once you have RNA, it's not so easy to work with this system. But in principle, that's like another thing to do, and definitely very, very interesting.
I have a question. So what are the requirements from the dimer, which is the basic assembly unit, to form a hollow capsid? I mean, if I just think about a particle that aggregates, it would aggregate and not be a hollow.
Right. So it requires the association between them to assume a certain curvature.
Yes, okay.
A narrow range of curvatures. And then to be strong enough, and it has 4 contacts. So it has 4 from the side, from the 2 sides, and from 4, basically 4 at the specific angles.
Okay, did you do, or maybe other group did a spontaneous simulation of such formation?
Mike Hagen, I think, did.
Okay, somebody or
and Roya Zangi, I think they made simulations that can also look at the other curvature and malformed particles, things like this. They looked at this actually. But the way we did it is we just took the final structure and we always kept intermediates on that specific position and orientation, and we either remove them or add them.
Okay.
So this was just to enumerate the possibilities.
Sort of a lattice model on the capsid.
Yes, on the geometry, the capsid.
Okay, there is about your conclusion that there are two-state model. I mean, you mentioned in your abstract Leventhal paradox.
Yeah.
It's also from the conclusion point of view, there is some similarity because in many cases, when you see protein folding, it is at least there is a significant amount that fold in a two-state. You can describe it by two states, unfolded and folded, especially with not with your protein, but one that have a native structure, defined native structure. You mentioned that it's not surprising. Yeah, I guess there is probably one can formulate a condition in terms of successive energy in your case about adding a monomer and a condition about the gain in energy versus the loss in entropy in order to make sure that there are no accumulations of intermediates. And okay, maybe in order to ensure in case of folding, rapid folding, and in case of, in your case, rapid formation of capsid, that is required to have it a rapid process. Otherwise, it's not work in nature. So that's why we see maybe
and also that there are no many local minimas that the system can be trapped in. In the aggressive condition, the reason we see intermediate is because there are local minima.
Yeah, okay, but maybe that's because you simply, you said that you increase the association energy or free energy when you integrate the solvent degrees of freedom. So that may be, one can think of a condition to put in to make sure that, or to guarantee that there are no accumulation of intermediates. I don't know if there are more questions. Okay, so thank you again for the nice talk.
Maybe there was a question.
Oh, there was a question.
I don't know. I have to check.
I
check it. Maybe in the Zoom.
Very good point on the energy gap,
not to have it too strong.
But I guess that's what the results show, actually, that actually for the thing to not be too stable or I mean, for the capsid to form, it's operating in a very narrow range of kappas.
Exactly.
It's very, very narrow because if it's too stable, then it goes down. And
exactly.
And if it's
that it will not form.
Yeah, and it was I mean it's very subtle. So I guess that viruses have to evolve very within
biology would need to be.
Yeah, so it's a very narrow range of possibilities, for viruses to remain efficiently putting themselves together, I guess.
I don't see anything.
Okay, so there are no questions.
Well, it's fantastic. Really nice.
Now we need to somehow
So
Yeah, normally I supposed to do it from here, but I disconnected because it was just giving the talk, what we're saying, it was here, you know.
yeah, so is he visiting for a few days?
No, I don't know if you want to talk with him. It's
Yeah, maybe a bit tricky today, but
okay.
yeah.
Yeah, with all the situation, I did not want to.
No.
No, well, with things going around, I did not want to
Yeah.
have a separate announcement and ask, ask for.
Yeah.
I was just telling him, didn't want to
There's too much.
waking up.
Yeah, no, but that was fantastic. I enjoyed so much the work. Yeah, really enjoyable stuff. Yeah. So anyway, Ronen,
Well,
fantastic
thanks for
to.
coming.
Yeah, no, it's so much. I learned so much.
Me too. Well, actually, me too, because I did not know
No,
you.
it's super cool work. Yeah, really very enjoyable. So yeah, I guess I'll see you around.
She's using our program.
That's
Yeah.
Paula. Yeah,
Yeah, that's
Paula.
Yeah. There's a good, well, I don't know if it's WAX or SAX community around in CFM, no, in the Neighbor Institute.
Okay.
Yeah. Anyway, guys, see
Thank
you.
you.
Yeah, bye-bye.
Yeah. Maybe there's one
Hmm?
who is using the same software, your software.
Yeah. They're trying to use the same substance.
Yeah, so it's famous. I mean,
Yeah, it's open to the public.
it's
open source.
open source.
You can, I should have said it, but it's an open standard. I put the website. It's no one of the website. Oh, very good.
Will you leave today?
Yeah, it's
Tomorrow.
tom-
Tomorrow. Go tomorrow. Apply tomorrow. Back home. Yeah.
So where are you now?
we probably will go to eat. If I mean, if we can if tomorrow if we tomorrow I think it's it's okay with me. I don't know, in the morning. Tomorrow. Yeah, so whenever,
Thank you. And nice to see you. Nice to meet you. How are you? Say how you did you like okay, relax. Very proud. Bro, here. This is hot. I still want it. It's cool. Congrats. Thank you. Yeah, come on, that's true. I got try. Yeah, you work here. Yeah, work here. Yeah. It's a bit beneficial. Yeah, for the defeat. Yeah, I know you had it. You had it. Yeah, it's fun. I mean, I didn't win well, so okay. How about you?
Me fastidia un poco porque también me saca mucho más con la teoría que hay que ver. Todo como y que haga yo entero. De la cabeza. No, de la cabeza.
Carlos, perdona que te moleste, ¿dónde andas? Ah, vale, vale, joder. Vale, vale. Cuidado con esto, ¿eh? De nucleo focal de la fuerza. Sí.
Vale, esa, gracias, gracias a ti. Muchísimas gracias, te hago un favor enorme. Gracias. Venga, pues. Gracias.
Hola, Gustavo.
Aitor,
Eso es.
¿qué tal?
Gustavo.
¿Cómo estás?
A ver,
Dame un segundo porque me falta otro chico que tenía que venir conmigo, que
Claro.
pasé por el despacho y no
ok.
le voy a escribir a ver si, no sé si estaba viniendo.
Y la voz no sabía de quién venía.